Yes, today we are going to start with the protozoa, the phylum Protozoa. This phylum comprises of those types of organisms which are unicellular or acellular and eukaryotic. So, what do you mean by unicellular or acellular? That is, it is having only single cell, and eukaryotic means it comprises or it consists of membrane bound organelles. Membrane bound organelles such as mitochondria, Golgi apparatus and true nucleus. So these are having unicellular, that is single cell, and membrane bound organelles. These are known as protozoa. So protozoa, when we split proto and zoa, when we split protozoa, proto means primitive. Proto means primitive and zoa means animals. So protozoa, primitive animals. So these are basically called animals because of heterotrophic type of nutrition. That is, they are having heterotrophic type of nutrition. That is, they will depend on other animals. They cannot photosynthesize. Except some examples. These are simplest animals. Simplest animals. That is, they have protoplasmic. They have protoplasmic grade of body organization. Body organization. That is, the cell is having the protoplasm and some of the organelles is found in the protoplasm. No other system or no other organs are found. So these are having protoplasmic grade of body organization. That is why these are known as simplest animals or simplest organisms. Now, these simplest animals may be free living or they may be parasite. That is, what are parasites? Those which absorb nutrition from living organisms, that are known as parasite. So they may be free living or parasite. And most of the free living protozoans are aquatic. That is, they are found in water, whether it is fresh water or marine water. Now, other characteristics involves these primitive animals. Other characteristics involves locomotion. How they locomote. Locomote means how they change their position. So locomotion with the help of different types of organelles such as flagella. Smaller ones, the longer ones are flagella. Smaller ones and more numerous in number, cilia, and a typical type of false legs called pseudopodia. Flagella, cilia or pseudopodia. With the help of these organelles, it can locomote or it can change their position. Locomotion. Now, what about the nutrition? As we have seen that they can be free living or parasite. So they can be holozoic nutrition or parasitic nutrition. That means when it is free living, or if it is not parasitic, or this will not depend on the living organism, then they can engulf whole organism. That is type of holozoic. Animals are engulfed. Holozoic or holophytic type of nutrition. When there is engulfing the plant, then it is called holophytic, otherwise holozoic nutrition. Now, holophytic nutrition, holozoic nutrition and parasitic nutrition. Now digestion, as far as digestion is concerned, as far as digestion is concerned, it is intracellular or vacuolar. That is, they can form the food vacuole. They may have the food vacuole with the help of phagocytosis, or with the help of pinocytosis. It can engulf the liquid particles. So it can be intracellular or intravacuolar. The pH of the vacuoles, may be first acidic, then alkaline. First acidic, then alkaline. The pH of the vacuole will be first acidic, then alkaline. You must have remembered about the digestive system of human beings or the mammals. The condition or the environment in the stomach is acidic, then in the intestine it becomes alkaline. So there is a mixture of the processes in the same vacuole. It becomes acidic, then alkaline. So it may be comparable to the digestive system of the higher animals, where there is acidic environment in the stomach and alkaline environment in the intestine. So digestion, as far as digestion is concerned, it is intracellular, that is within the cell and within the vacuoles, and pH becomes acidic, then alkaline. Now the digestion, if it is digesting something, it should excrete. So as far as excretion is concerned, there is no special type of organ for excretion. There is no special type of organ for excretion. So excretion takes place by plasma membrane. Excretion by plasma membrane, as well as respiration is also by plasma membrane. Respiration is also by plasma membrane. So locomotion, excretion, respiration, it is performing all the functions of a normal animal. That is why these are considered as animals. And at the time of reproduction, it can reproduce with the help of fission. It may be binary fission or it may be multiple fission. Binary fission or multiple fission. Along with this, they can also reproduce with the help of budding also, and a similar type of sexual reproduction also. So they can reproduce asexually as well as sexually. Now, at the time of unfavorable condition, So all the things are going to be under favorable condition. But if there will be unfavorable condition, then what they do, they will parinate. They parinate with the help of the formation of a hard covering over their surface called cyst. So it prevents them from desiccation and the unfavorable condition outside the body or the environment. So they can parinate. Parinate means they then continue their race with the help of encystment, and when favorable condition comes, they can parinate or they can again rejuvenate and forms new organism or with the help of reproduction. So these are the general characteristics of phylum Protozoa. So once again we have to see. Now we have to see what are the main characteristics. They are the simplest organism. These are the simplest organism. They are covered by cell membrane. They are covered by cell membrane or pellicle. Third thing is protoplasmic type of body organization, locomotion by flagella, cilia and pseudopodia. As far as nutrition is concerned, it can be holozoic or holophytic or parasitic, and excretion and respiration through general body surface. Through general body surface, and they form cyst during unfavorable condition. So these are the main characteristics of phylum Protozoa. Now we are going to classify them on the basis of locomotory organelle. As we have seen that they change their position from one point to the next point with the help of some organelles or different types of organs. So these basically, the protozoa is divided into different categories on the basis of their locomotory organelles. So these are divided into subphylum Plasmodroma and Celiophora. Now this Plasmodroma is again divided into different categories. These are subphylums, and these are classes. Number 1, Mastigophora. Then we have Rhizopoda. Then we have Opellinata and Sporozoa. Mastigophora, they have flagella as locomotory organelle. Rhizopoda have pseudopodia as locomotory organelle. This Opellinata also have flagella, but these are smaller than Mastigophora. Sporozoa have or do not or devoid of any locomotory organelle. And these class of Plasmodroma is basically endoparasite. Okay. Then we have Celiophora. Classes are Ciliata and Suctoria. The common examples: Mastigophora, Rhizopoda, Opellinata, Sporozoa. This is Euglena. Rhizopoda with the help of Pseudopodia, that is Amoeba. Opellinata, that is Opellina, and the example of Opellina, and this is Plasmodium. Ciliata, example Paramecium, Suctoria, Ephilota. So the whole phylum is divided on or categorized on the basis of locomotory organelle, Plasmodroma and Celiophora. Mastigophora, Rhizopoda, Opellinata, Ciliata and Suctoria. Now this is the complete classification of phylum Protozoa. Now we will study about some of the members of phylum Protozoa. Phylum Protozoa have different types of organisms, and most and very much interesting type of organism is Euglena. Because it is having characteristics of both animals as well as plants, and they are performing the mixotrophic nutrition because it can photosynthesize also, and it can also absorb nutrition just like animals. So this type of nutrition is known as mixotrophic nutrition. So let's have a look on the general structure of Euglena. This is also known as sleeper animalcule. And it is having two flagella. Not two types, two flagella. One short flagella and one large flagella. This is not having any type of cell wall. It is having the pellicle. These have myonemes, and stored food material is paramylum bodies. These are paramylum bodies. Here you will find an eye spot, and this part is known as the cytostome. Now we are rubbing it because there is a loss of position. This is known as cytostome. It is comparable to the mouth of the organism. And this is the cytopharynx, comparable to the gullet. So this very Euglena is performing mesotype of nutrition. It does not have any cell wall and it is surrounded by a plasma membrane called, it is performing by a plasma membrane. Plasma membrane, that is known as pellicle. So this organism is going to perform dual function. It can photosynthesize and stored food material is paramylum bodies. Now next is our amoeba. Next is our amoeba. This organism do not have any specific shape. Do not have any specific shape. These do not have any specific shape. So let's have a look on the structure and its organelle. It moves with the help of pseudopodia. Say this is pseudopodia. This is the nucleus, a true nucleus. Then we have food vacuole and a contractile vacuole. This is the contractile vacuole. This is contractile vacuole which helps in the osmoregulation. This is ectoplasm, and in the middle you will find endoplasm. Ectoplasm and endoplasm. So this organism do not have any specific shape. It can be modulated into different types of shapes according to its need. So this pseudopodia, that is false leg, it moves or it locomotes with the help of pseudopodia due to sol-gel hypothesis. That is, if gel converts into sol and sol converts into gel to form pseudopodia, and with the help of pseudopodia it can change its position or for the capture of the food or for any other purpose. So amoeba is having no spatial type or no fixed shape and size. This amoeba proteus can reproduce. How they reproduce? This amoeba can reproduce or it can increase its number with the help of binary fission. Then what we have? Multiple fission. Then we have sporulation. Binary fission means when the cell divides into two daughter cells. Then this is known as binary fission. Two daughter cells. Then it is known as binary. Bi means two. In multiple fission, it can form several or more than two. Several or more than two cells. Now sporulation by the formation of special types of spores. But one thing should be in mind, that binary fission takes place during favorable condition. During favorable condition. This takes place during favorable condition, and this both takes place during unfavorable condition. So binary fission, sporulation and multiple fission, it can perform different types of reproduction with the help of binary, multiple and sporulation. But binary fission takes place during favorable condition, and sporulation and multiple fission takes place during unfavorable condition. So as we have seen that this do not have any shape, so it moves with the help of pseudopodia, with the help of sol-gel conversion. Now this amoeba was basically discovered by Russell von Rosenhoff. Russell von Rosenhoff on a basis of a Greek god, that is amoeba. Russell von Rosenhoff. Now after amoeba and different Euglena, we are going to have, or we are going to study about a very notorious member of this protozoa is Plasmodium. 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 This Plasmodium is notorious. Why it is notorious? Because it causes a deadly disease. It causes a deadly disease which becomes epidemic or endemic disease. That is called malaria. That is called malaria. So malaria is responsible for manifold of deaths in developing countries still. So it is curable, but the drugs are not in the reach of some of the very backward, or you can say Saharan region or African countries. So it is more prevalent in African continent. So the Plasmodium is causing the disease, malaria, and now it is a digenetic parasite. It is a digenetic parasite. What do you mean by digenetic parasite? That it completes its whole life cycle in two organisms. That is why it is called digenetic parasite. So it completes its whole life cycle in Anopheles, or rather female Anopheles mosquito, female Anopheles mosquito, and in man. In man. It is the primary host and this is secondary host. Primary host and secondary host. Now in humans, it is a digenetic. So in humans, asexual part of the cycle, asexual part of the life cycle prevails. But in case of Anopheles mosquito, sexual part prevails. Humans asexual, Anopheles sexual life cycle. So Plasmodium is a notorious type of organism which causing a deadly disease that is called malaria. It is a digenetic parasite. It involves humans and Anopheles and completes its life cycle in both of them. And humans are also acting as intermediate host. That is, it can act as a reservoir. Reservoir means what? It can have the stock of the sporozoites. Sporozoites, that is its infectious term or infectious stage of the Plasmodium. Now we are going to see what will happen in the humans. What will happen in the humans? In humans. So when a female mosquito bites, a female mosquito Anopheles bites a human, and if it contains a sporozoite stage, it inoculates a sporozoite stage in the humans. But not every Anopheles, those Anopheles which have in the salivary gland, those sporozoites are inoculated in the human beings. Then what will happen? Liver schizogony. Liver schizogony. That means the sporozoite, this sporozoite will enter the hepatic cells. Hepatic cells means liver cells within half an hour. After entry of the half an hour, it goes directly towards the liver and enters the liver cells, that is hepatic cells, where they divide or converted into the sporozoite is converted into cryptozoite. Cryptozoite. Sporozoite is converted into cryptozoite. So in humans, female Anopheles inoculates sporozoite. So for humans, the sporozoite is the infective stage. This is the infective stage. Infective stage for humans. But here one thing should be in mind, not every Anopheles can inoculate. Only those Anopheles which have in the salivary gland the sporozoite can cause infection to other organisms. Then this sporozoite is directly goes towards the liver, because liver is the most busiest gland and it performs different types of functions. So it is going directly towards the hepatic cells or the liver cells, where these sporozoites changes into cryptozoite. These sporozoites are converted into cryptozoites. Now these cryptozoites will multiply by fission, breaking, and forms cryptomerozoites. These cryptozoites multiply by fission, then forms cryptomerozoites. Now these cryptomerozoites will be converted into different stage. The cryptomerozoite will be converted into phanerozoites. And this will be converted into micrometa cryptomerozoites. Micrometa cryptomerozoites 64 and macrometa cryptomerozoites about 1000. Micrometa cryptomerozoite and macrometa cryptomerozoite. This cryptomerozoite is formed from cryptozoites. This will be converted into phanerozoites, and this phanerozoite will be converted into micrometa cryptomerozoite and macrometa cryptomerozoite. This stage is known as exoerythrocytic schizogony. Exoerythrocytic schizogony. Exoerythrocytic schizogony. Now this will again starts new cycle, and this will undergo signet ring stage. Signet ring stage. This cryptomerozoite to phanerozoite, phanerozoite to micrometa cryptomerozoite, then macrometa cryptomerozoite, then it will each of them will form new cycle, and this each of one will undergo signet ring stage and becomes amoeboid. Amoeboid, that is becomes irregular in shape. There is no definite shape. That is known as trophozoite. This is known as trophozoite. This is known as trophozoite. So this will be converted through the stages until trophozoite, amoeboid trophozoite. Now next stage is schizogony. Erythrocytic schizogony. Erythrocytic schizogony. So the amoeboid trophozoite. What will happen to the amoeboid trophozoite? That means cryptomerozoite will be converted into macro and micro cryptomerozoite. That will be again converted into the amoeboid trophozoite. This amoeboid trophozoite will be converted into schizoids, and again by dividing multiple fission, they will cause multiple fission undergo and forms many schizoids. Next stage is schizoids. Amoeboid trophozoite will be converted into schizoids, and these multiply many times with the help of fission, and they forms schizoids. And these schizoids will rupture the RBC. RBC ruptures and released into the blood. This is released inside the blood, and this is the stage when there is severe shivering as well as persistent increase in the temperature of the body takes place. That is called fever, along with the chill or shivering takes place when cytosol becomes released in the blood. So this amoeboid trophozoite will be converted into cytosons, then into cytozoids, then it will rupture the RBC and released into the blood. And when it is released into the blood, it will cause severe chill as well as fever. So all these cycles from sporozoid to the cytozoid stage, all this takes place in the human beings. That is the asexual part of the life cycle of the Plasmodium. Now, these cytozoids are taken up by the Anopheles mosquito. So now life cycle in mosquito. That is life cycle of the Plasmodium in mosquito. So the cytosol stage released in the blood of the patient when it is bitten and sucked up by the mosquito, it takes out cytozoids, and these cytozoids will undergo gametogony. That means this will form two types of gametes, that is microgametes and megagametes. These cytozoids will undergo gametogony in the mosquito. This stage is sucked up by the mosquito. Then they will undergo gametogony, microgametes and megagametes. That is male gamete as well as female gamete. This is 6 to 8 microgametes are formed and only one, I repeat, only one megagamete is formed. Only one megagamete is formed. These are microgametes, these are megagametes. So microgametes as well as megagamete, this microgamete formation is called exflagellation. Exflagellation. So under gametogony, microgametes, that is by the process of exflagellation, 6 to 8, 6 से 8 exflagellation microgametes are found, and megagamete, only one megagamete is formed. So male gamete as well as female gamete is found. So it is the sexual life cycle or sexual part in the mosquito takes place. Now these micro as well as macro or megagametes. Now micro gametes and megagamete will fuse or fertilization takes place after 10 hours of entrance. This is known as fertilization. What? Fertilization takes place and forms zygote. So now this zygote will form eukinete. A next stage that is called eukinete. This eukinete will enter wall of the stomach in mosquito, wall of the stomach, and becomes encysted. That is known as oocyst. It becomes oocyst, and this oocyst will undergo division, multiple division, and forms sporozoites in the sporon. This oocyst is also known as sporon, and these are liberated or transported to the salivary gland of the mosquito. So when it reaches the mosquito's salivary gland, it is now ready to be inoculated in the different organism, and this sporozoid is the infective stage for the human being as well as for different types of animals. And the stage which the mosquito is sucking is the schizoid stage. So this complete life cycle of Plasmodium, which is causing a very deadly disease, or which can cause or which is causing lots of deaths in the developing countries as well as in the African continent, that is called malaria, is a digenetic parasite, and it converts, or it is going to convert the whole population into the minor population by causing the death of the people. But this can be curbed by the use of different types of medicines and with the help of preventive measures. So this malaria, if not treated well, or if not going to be